Hey everybody, welcome to SoMAS's virtual recruitment event. My name is Chris Paparo and I'm the manager of the Marine Sciences Center. The Marine Sciences Center is located at our Southampton campus, which is about an hour drive from the main campus. It's located in the town of Southampton and it's on Old Fort Pond, which is a small bay attached to Shinnecock Bay, which is the easternmost large bay on the south shore of Long Island. This is home to our Semester by the Sea program, which is an undergraduate program where students are immersed in the marine environment. All their classes are in this building. All their labs are on our research vessels. They can walk up the block to get into the water to do seining, snorkeling, etc. We also have a conference room overlooking Old Fort Pond. This is a great place for meetings, conferences. We can accommodate up to 20 people in this room. But the real gem of the facility is our wet lab. So we have a marine wet lab where we are able to have three different seawater scenarios. Then the first one is straight up raw water, raw seawater that gets pumped in from Old Fort Pond. It then goes through these pumps, goes into the lab, and feeds the various sea tables. The water leaves those sea tables and goes back to Old Fort Pond. Now that works great for our researchers that are studying shellfish, which is probably about 90% of the work that goes on in this lab. By using raw water, we don't have to grow as much algae to feed them. We also don't have to do water changes because the water is constantly flowing through. So you always have clean, fresh seawater full of phytoplankton, zooplankton, and other food items for the animals that you might be raising. The problem with the raw line that we have is anything that happens in Old Fort Pond can also happen in the lab. The first thing we see is temperature. So like this time of year in the dead of winter, right now Old Fort Pond is just above freezing. It has froze in past years, actually it froze for about a week this season as well. With a temperature of 0, there's not a whole lot of research that can take place. So we have a temperature control line where I can instantly heat or cool the incoming raw water. Here is our heat exchanger. We use the building's boilers to heat up a loop of water. That's this white piping that goes into it. And we can run the cooler seawater in the opposite direction. So we'll have a counter current and we're able to heat that water at a rate of where I can bring it from 0 Celsius up to 20 or 25 is as high as I've gone at about 30 gallons a minute. So it allows our researchers to be able to work throughout the winter. In the summertime, the opposite happens. Old Fort Pond sometimes gets a little bit too warm, 25, 26, occasionally 28 degrees Celsius. While the average temperature in Shinnecock Bay is about 15, trying to replicate those experiments at 28 is difficult. So we have a chiller, that's this large blue item right here, that then is able to chill the incoming water. And I've been able to set up tanks outside in the sun without shade and bring the water from 28 down to 15 degrees Celsius. So again, we're able to do research throughout the year, no matter what the temperature is. The other problem we sometimes have with our raw water is we'll get harmful algal blooms that'll show up in Old Fort Pond. Typically late summer, we will see cochleodinium, which is the rust tide. Rust tide is harmful to shellfish. So if you're a grad student who's been studying a cohort of clams for say 3 years, and you're getting close to finishing, and in that 4th year, cochleodinium shows up and wipes out your clams, that means you're going to have to be with us for another 4 years. So we don't want that to happen to you. So we have a recirculating system. With this system, instead of the water going back out to Old Fort Pond, it leaves the sea table, goes through a bank of filters, a cartridge filter, biological filtration, foam fractionators, and then through a UV sterilizer before it goes back to the sea table. By doing this, we isolate the clams or the organisms from Old Fort Pond and any harmful algal blooms that might be there. The only issue we then have is we then have to grow food to feed them. So we have our own phytoplankton cultures. You can see a bank of 15 Caldwell tubes in the far back here. We raise about 8 different species of phytoplankton to be able to sustain some of the work that takes place here. So what work does take place here? Well, there's several researchers that utilize our lab. Chris Gobler probably is one of the most common users of the lab. His lab does a lot of work with ocean acidification and harmful algal blooms. Brad Peterson also does a lot of work at this lab, and the Peterson lab focuses on seagrass ecology. So everything from mapping current seagrass beds to seeding new seagrass beds to looking at predator-prey relationships that take place in those seagrass beds. And a lot of those field research trips, projects that they do where they're looking at those behaviors, we can actually also recreate here with mesocosms in the lab. So we're able to keep fish, crabs, shellfish, all at the marine lab. Some other stuff that takes place here is the Shinnecock Bay Restoration Program. Now, that's a grouping of several researchers working together. And some of the projects that that project has done is we've put out over 4 million adult clams, hard-shell clams, into Shinnecock Bay to help with these harmful algal blooms. We've also started our own oyster reefs, and as of now, we've had 4 oyster reefs out in Shinnecock Bay. We seed spat bags, which are pretty much just a mesh bag full of old clam shell or oyster shell that we set in these tubs. We seed juvenile oysters on them. Once they're about nickel and dime size, we then move all these bags out to the bay. One adult oyster can filter about 50 gallons of water a day. So with 4 reefs, each reef having over 1 million oysters, that's a lot of water filtration and helps of keeping the bay clean. Plus, it creates habitat for shrimp, crabs, fish, a whole wide variety of organisms that live in the bay. So again, that's some of the stuff that takes place with the Shinnecock Bay Restoration Program. We also have other researchers from main campus that come down and do quite a bit of work. Again, rotating through as grants come available or funding becomes available. Some of the other projects will change. As I mentioned, we are right on the bay, so that gives us easy access to getting out into Shinnecock Bay, the Atlantic Ocean, Peconic Bay. We have a fleet of vessels, as you'll see here. The RV Shinnecock is a pontoon boat that lets us get into really shallow water. Because it's an open deck, it's great for those oyster projects where we can hand bags over the side to plant them. It's also a great dive operation, so we can get on and off the boat, but this is strictly a bay boat. The RV Peconic is a little bit larger. With this, it's got some shelter so we can get out of the sun, especially for like the Shinnecock Bay Restoration Program. When they're doing their fish surveys, they're able to haul with the otter troll, bring their organisms on board and be in the shade while they collect and measure and sample their collection. The next boat we have is the RV Pominock, which is our ocean-going vessel. We're able to do otter trolls, beam trolls, deploy instruments. Lately, we've been doing a lot of work at deploying acoustic receivers along the South Shore of Long Island. So again, that gives us some great opportunities as well. Just as a reference on where we are, that blue dot is Old Fort Pond. That's the marine station. So here's Shinnecock Bay. This is the Ponquag Bridge. This is Western Shinnecock Bay. Here's Shinnecock Inlet. So pretty much 10 minutes, we're out into Shinnecock Bay. Give it another 15 minutes, we're out into the Atlantic Ocean. And 20 minutes, if we go through the Shinnecock Canal, gives us access to the Peconic Estuary. So again, our location is prime for doing various research throughout Long Island's marine habitats. Here's an overlook of Shinnecock Bay looking to the west. That bridge, like as I mentioned, is Ponquag Bridge. So for those that are scuba certified, that is probably one of the best places. Actually, it is the best shore dive in New York, and I would put it up there as one of the best shore dives in the Northeast. The diversity of wildlife you'll see at that bridge is amazing. Many of our grad students collect organisms there for their research. I personally do a lot of scuba diving and photography there. You'll see a lot of game fish like fluke, striped bass. You'll see blue crabs. Throughout Shinnecock Bay, we do have a lot of scallops. So if you dive and snorkel amongst the seagrass beds, you'll definitely see a lot of scallops. Seahorses are quite abundant throughout Shinnecock Bay, again, within our seagrass communities. And one thing that's kind of cool too is we also get a lot of tropical fish. It's just crazy for many people to think that, in the Northeast, we're seeing blue angelfish, bicolored damsels, butterfly fish, triggerfish, but we get a lot of really cool stuff that drift up here in the summer months. If you're into larger fish, we get a lot of sharks locally. This is a blue shark that was tagged and getting ready to be released. We also see mako sharks, thresher sharks, sand tiger sharks, duskies, brown sharks, tiger sharks. This was one that I photographed a few months ago back over the summer. Hammerhead sharks. So, we get quite a diversity of sharks, even white sharks. This is a juvenile young of the year that me and my colleagues had tagged off of Montauk 2 seasons ago. So if you're into sharks, we do have a lot of that here in our area. Maybe you're into wildlife. We have a lot of cool wildlife, especially this time of year in the winter. We get some really cool ducks. These are hooded mergansers. Bald eagles are super abundant these days. It seems like I could see one almost every day while I'm working at the Marine Station. Snowy owls. This was at the Barrier Island, which is about a 10-minute drive from the college. We get these winter visitors every year from about October to about March. Osprey, if you're into fish hawks, this is right outside the window of the Marine Center. This is our Osprey platform. This is mom with her two babies. They were about 2 weeks, 3 weeks old, I think, here in this picture. Maybe you're into marine mammals. We have a lot of them out here as well. Harbor seals are very common from about October through March. We also will see gray seals, harp seals, and occasionally hooded seals and ring seals. Maybe you're into bigger marine mammals. We have a lot of dolphin. These are bottlenose dolphin. Again, you can see how close to shore they are. And we get a lot of whales, humpback whales especially, close to the beach. One of our researchers, Leslie Thorne, she has a couple students that work with us at our station where we take them out on our 25-foot Parker, where they're IDing and mapping, or cataloging these whales that are in our areas through photography, drone photography. So again, it's they go out quite a bit when the weather permits. But you can see they're right here close to the beach. So even if you're not studying whales, it's just a great way. You can go down to the beach and see a whale lunge feeding on bunker. There's the Ponquag Bridge that I mentioned in the background. And there is, again, the Ponquag Bridge at sunset. So I hope this was informative for everybody. If you have any questions, please feel free to reach out to me. My name again is Chris Paparo. My email is at the bottom of the page here, christopher.paparo@stonybrook.edu. Again, sorry we couldn't do this in person, but hopefully you'll accept and come to SoMAS and we can meet in person. I can show you all this stuff in person. So again, thank you so much. Enjoy the rest of the event. Thank you.